Good evening. STEM Minority here, and tonight I'm going to talk to you guys about um, how to read research papers, and more specifically, we're going to focus tonight on what to get from an abstract and how to read an abstract. I think last time we left off, we left off the introductory video. I told you guys that this channel will be based on, helping you guys try to understand how to read a research paper, and hopefully through watching these videos you'll find it easier to understand how to read a research paper, and in doing so you'll find science, if not fun, then more tolerable. Alright, so first things first, we're going to focus on how to read abstracts. Alright, and then later on we're going to focus on, or I'm just going to talk about, some of the latest trends in science using the techniques that I show you. All right. So, how to read an abstract. So, when you're reading an abstract, or I'm going to first start from the beginning. When you're reading an abstract, you're doing so from a certain point in your research. You're doing it from like the first preliminary stages. And in the preliminary stages, or in the first stages of your research, you're not really trying to delve deep into reading the research paper. If anything, I think maybe if you guys watch other videos on how to read abstracts, a lot of what they'll tell you is, when reading a research paper, the first thing you do is skim it. You don't really read to digest all of the information, and they're right. You only get a rudimentary understanding of what the researchers were trying to do. So at this point in the research, all you're trying to do is gather papers that could be related to your topic of interest. So you've developed a topic of interest, or you've done, and in a sense, this is your background research. You are getting information that you could use towards a research topic. And or if you don't have a topic of interest that you listed down, then reading the abstracts is a good way for developing an idea for what you want to do. So let's say I wanted to make this this go green technology. Or let's say at first, going into researching, I did not know how to, or I didn't know what I wanted. I knew I wanted to make this go green technology, but I didn't know what go green technology I wanted to make. Well, reading abstracts is a really good way for helping me develop an idea. And after reading abstracts, you might say, hey, I want to make a new solar panel, or hey, I want to go into a new form of energy harvesting. So reading abstracts is really good for developing ideas. And lastly, even if you're not trying to develop an idea or gain papers related to your idea, it's good to read the abstracts to see just how far research has gone in your field of interest. For example, recently I was studying REWAD, which is reverse electrowetting, and that's a new energy harvesting technique that somebody came up with, and it got kind of far, but I'm still reading into it, and reading the abstracts is a really good way to sort of gain a baseline knowledge about your field of interest. So in a sense, again, you're either gathering papers that are related to your topic of interest, you're developing an idea for yourself about the project you want to pursue, or you're just simply educating yourself. And in reading the abstract, you will answer these questions. All right, they're not in-depth questions. They're really basic questions. What did the researcher do? All right, the researcher will say, well, after observing this, we did this, this, and this, and we came up with this conclusion. And it's really simple. So what did they do? What did they expect, and what conclusion did they reach? And in a sense, you're just gaining a really basic understanding of what they did. they'll tell you what they were experimenting with. They may have been experimenting with bacteria. They may have been experimenting with mice, with Hydra, and they may have been experimenting with maybe their genes, with gene regulation or something like that. They were doing something. And then they may even drop what techniques were used. They may have said, oh, we did a Western blot, so on and so forth, But the whole point is you're getting a basic rudimentary understanding. Okay? Now, before we dive into, reading our first abstract, there are you can dive in with two sort of mindsets. All right? Now, some of us that are a little bit more experienced, and we've been in the field for a minute, we'll dive in with sort of the research topic that we have in mind. So we'll dive in, like, okay, for example, I might dive in and I might go, okay, what are some, there are bacteria that, are good for helping, are bacteria that are really good for bioremediation. Okay, so I'd be focusing on papers that focus on bioremediation and the latest trends in bioremediation. Okay, so I'd be looking for papers that matched bioremediation, and that's it. Okay, and I try to see, and I'd already have some sort of topic in mind that I want to do, and I try to see if the papers matched my or were in line with what I wanted to do, so I could basically have a strong understanding of my topic, right? And so I could also I guess have background supporting information for when I write those first few paragraphs, and if you don't know what you're doing, which is probably going to be 99% of us, let's be honest. If you're just kind of getting into researching, like you're a not a high school freshman, a freshman in college or something, then really your main focus at this point would be to sort of just develop, just to kind of narrow down a field. If you're interested in biology, just go, hey, I'm interested in biology. If you're interested in bacteria, just say you're interested in bacteria and just read papers on bacteria and just try to understand the abstract as best you can. And if you run into any things where they say, hey, we did this, this, and this, try substituting the words for, for some words that you do understand so that you get a gist of the paper. All right. So that being said, let's go ahead and dive into our first abstract. So the database I like to go to is NCBI PubMed. I go there all the time, right? And you can type in your topic of interest into the search query. So we're going to type in bacteria and bioremediation, like I said before, right? I know y'all might not be interested in that, but that's what we're going to do. So we can go ahead and, we're just going to go ahead and do that, right? Let's see. And I guess we can use this right here. The title, you want to focus on the title. Let me go back real quick. You want to focus on the title because the title sort of tells you, the beginning and the end of this, research paper. It tells you everything. Neustonic versus epiphytic bacteria of eutrophic lake and their biodegradation ability on deltamethrin. You don't really need to know these terms. All you need to know is you got two types of bacteria, Neustonic versus epiphytic. I don't know what that means, and I don't know what that means, but there are two types of bacteria on a eutrophic lake. So this lake is affected in some way. I know what eutrophic means, but for those of us that are just really new at this, just understand that the lake is affected in some way. and their biodegradation. Hopefully you know what degradation is. You're degrading something. The ability to degrade delta methrin. So we have they're measuring two types of bacteria on a lake that is affected, and they want to know if they can degrade this chemical called delta methrin, which I assume is bad. I wouldn't know. I mean, I would know, but I'm looking at it from y'all's point of view. All right. So just to give credit, this is the authors. I'm just gonna say their first names: Angiska, Jacek, Iwana, and Wajzish. Oh God, I hope y'all don't see this video. That'll be tragic. So let's read together. This study evaluated biodegradation of the insecticide deltamethrin. Okay, so first thing we know is deltamethrin is an insecticide. It kills insects. Okay, this is the concentration by pure cultures of. So the study evaluated biodegradation of insecticide deltamethrin by pure cultures of nistonic and epiphytic bacteria and by mixed cultures. So they're measuring the degradation of this by this kind of bacteria and this kind of bacteria, and when they're mixed together, which consisted of a mixture of 25 bacterial strains isolated from the surface microlayer, so isolated from some area, and epidermis of the common reed, growing in the littoral zone of eutrophic lake. I'm not going to pronounce that. So they basically pulled this bacteria out of whatever this is. So results indicate that whatever this is and whatever this kind of bacteria is are characterized by a similar average capacity to degrade deltamethrin. So they found that both kinds of bacteria can degrade deltamethrin at some rate. After a 15-day incubation, bacteria isolated from the surface microlayer reduced the initial concentration of deltamethrin by 60%, while the average effectiveness of the bacteria found on the common reed equaled 47%. Okay, so let's reword the paper. There is this insecticide, deltamethrin, in this lake, in this eutrophic lake, and they measured two types of bacteria and a mixed culture from the common reed to see just how much it can degrade deltamethrin in this eutrophic lake. And they did a 15-day incubation in, I guess, water collected from that lake or simulated to sort of represent that lake, and they found that it reduced the concentration of deltamethrin by 60%. I know that sounds a lot, but it's really simple. You have a bunch of bacteria. They wanted to measure the degradation rate of this chemical or this insecticide, and they found that bacteria and they, I guess, measured it, and that's all they did. Okay? So that's what you take away from this. This is what they did, all right? And then you just kind of keep it going. Honestly, that's what I do. Snap. Hi everybody, I'm back. I don't know why the video, for some reason the video cut off at a certain time, but I'll pick up where we left off. My bad. The editing's going to be hella funky though. Or I shouldn't use that word. It's going to be really funky. But yeah, we were just talking about the deltamethrin, and again, it was really simple. They answered these questions, and all you really need to do is, again, if you don't know what's going on, substitute the higher tier words for terms you'll find easier to understand. Again, this is the first reading, the first time around, so you honestly don't need to get an in-depth understanding of what's going on. Again, it's not that deep. It's not that deep, okay? And for those of us that are experienced, just keep in mind, does what they're doing match with what we want to do? This is for the more experienced of us, does it match? For those of us just entering, can you understand it? Just substitute the vocabulary for words you do understand, okay? Now, Just making sure the time's still going, my bad. Okay, now there's another paper. I already read it when the video shut off, so I'm going to read it again. Regulation of coastal methane sinks by a structure gradient of microbial methane oxidizers. All you're really doing is just, okay, now keep in mind, substitute the words for words we do understand. You're regulating, I guess, methane by some structure of bacteria that oxidizes the methane. Okay, so that's what you're doing. You're regulating methane by bacteria, or by some structure of bacteria that oxidizes methane, or some method of bacteria that oxidizes the methane. And you can assume that by oxidizing the methane, it helps get rid of methane. Okay, so coastal wetlands are widely recognized as an atmospheric methane source. They got a lot of methane, or the atmosphere has a lot of methane in coastal wetlands. That's easy to interpret. However, recent field studies suggest that some coastal wetlands could also act as methane sinks, so they could help methane disappear, but they don't know what's going on. The mechanism is not yet clear. Here we investigated methane oxidation with different electron acceptors in 4 coastal wetlands in China. So they tested from samples in China using a combination of molecular biology methods and isotopic tracing technology. So they took these molecules, which you can find on the periodic table, and they oxidized methane, and they tested on 4 samples in China using these methods. You don't need to know the methods, they just used these methods, okay? The geochemical profiles and in situ Gibbs free energies, so you can interpret that as the analysis suggests that there was significant nitrite-dependent anaerobic oxidation of methane. So there was anaerobic, which means there was, at this point, I'm assuming you don't know what anaerobic means, so there was this kind of bacteria, or this style of oxidation of methane, and it was nitrite dependent, so you need nitrite to do it. I don't expect you to know what nitrite is, just understand that whatever it was nitrite dependent, and it was this style of oxidation of methane in subsurface sediments, so below the surface sediments. So there was no oxygen down there. This was subsequently experimentally verified, so they just verified the stuff. Remarkably, the methonotropic community seemed to exist in the sediments as layered structures, and the surface aerobic methane oxidizing bacteria were able to take up atmospheric methane at a rate of this. So basically, the aerobic methane on the surface, so you can assume that aerobic means that they use oxygen, so aerobic methane oxidizing, this is the style. So the anaerobic oxidation means that, they don't use oxygen to oxidize the methane, or they can't use oxygen. They're anaerobic, whereas this is aerobic, so they do use oxygen. So the methane oxidizing bacteria took it up at this rate, and the sedimentary methane was completely consumed by anaerobic methanotropes, so non-oxygen, which means subsurface methanotropes, or this kind of bacteria, they took it up at this rate. These results suggest that coastal methane sinks might be governed by a diverse microbial community. So it might be, the degradation of methane, or the disappearance of methane, might be caused by, I guess, a bacteria in groups. That's all you need to know about diverse microbial communities where NC10 methane oxidizers contributed significantly. This finding helps to better understand and predict coastal methane cycle and reduce uncertainties in the estimation of global methane flux. So this study helped with this effect, helped predict the methane cycle. So where does methane go? Where does methane come from? But more importantly, where does it go? Right? Helps predict the methane cycle, and it helps reduce estimations in that understanding of the cycle. Okay? So in short, remember, you're in the beginning stage, so you're just gathering papers that you're interested in, And these are the questions that the researchers are, I guess, presenting you with. And then if you're an experienced researcher, just keep in mind, does it match your topic of interest? And then if you're not an experienced researcher, if you're just getting into it and you want to get kind of hooked onto it, again, just substitute words that you don't know for words that you do understand. If they talk about some method, just call it a method. You don't need to call it the whole thing like the shabalaba something, all right? Just keep it simple, and you will gain a better understanding of it when you go through it the next time around. So when we come back, we'll go back to NCBI, and I guess that's when I'll start going over the first few papers and advances in biology and science. And I'll do the same method with reading the abstract, and hopefully y'all follow along. And yeah, I'll just, go through another abstract or two tomorrow, and I'll post that video tomorrow. So hopefully y'all understood what just happened, and if you like it, please leave a like. If you would like better clarity or understanding, just indicate in the comments. If I'm wrong, please indicate in the comments if I'm wrong about certain details, because again, I'm trying to basically dumb down papers for your, people who don't understand science to understand. Okay, so yeah, please leave a like if you enjoy, and I'm out.